Very good morning. Today we'll talk about electrophoresis. This course is for second year BTech Biotechnology students. The aim of this session is to familiarize students with the basic concept of electrophoresis. The objectives of this course is this session is especially designed to remember electrophoretic systems, understand process of electrophoresis, apply the electrophoretic method for analysis, and analyze biological samples. The learning outcomes of this session is to remember various types of electrophoresis, summarize various types of electrophoresis, and apply the role of the gels in separation of various biomolecules. Coming to the principle, electrophoresis is one of the important bioanalytical tool in academic research, diagnostic research, pharmaceutical labs, forensics labs, food and agricultural research facilities. It has a wide scope of application. Now let us see what is electrophoresis and what is its principle. Electrophoresis is a method used for the separation of charged molecules from one another based on differences in the migration field. When this is subjected under an electric field. Now molecules which can be separated based on their any physical property, those molecules can be subjected under electrophoresis and they are subjected based on their migration speed. Coming to introduction, in electrophoresis we use two electrodes which are made up of an inert metal like platinum that is immersed in two separate buffer chambers. So if you imagine a gel apparatus, there will be two reservoirs where we can load our buffer. The two buffer reservoirs will be quite opposite to each other, and these two are fully isolated from, they are not fully isolated from each other. Charged particles can migrate from one chamber to the other. Now when you apply electric power, this is generated between these two electrodes. Now there will be two electrodes, one is cathode, the other is anode, where the buffer flows from one to the other, makes the molecules to get separated. Now coming to the types of electrophoresis, electrophoresis is broadly classified into two types. One is free electrophoresis, the other is zone or zonal electrophoresis. Under free electrophoresis, there are two different types. One is microelectrophoresis, the other is moving boundary electrophoresis. However, the choice of free electrophoresis is absolute because nowhere this is being implemented, because there are certain limitations in the choice of free electrophoresis. However, they are traditionally done. One is microelectrophoresis, the other is moving boundary electrophoresis. Coming to the second type of electrophoresis, which is called as zone electrophoresis or zonal electrophoresis. They are of three major types. One is paper electrophoresis, second one is cellulose acetate electrophoresis, and the third one is gel electrophoresis. This is the type of electrophoresis which is very much important and being used till date. There are several different types of gel electrophoresis which we will be discussing in the subsequent slides. Now let us talk in brief about the free electrophoresis, although this is not being widely used. One is microelectrophoresis, the other is moving boundary electrophoresis. Now you can see there will be no stabilizing medium in either micro or moving boundary type of free electrophoresis. How the process is carried out is, a buffer will be chosen and the sample will be applied directly to the buffer, which means the sample will not load at a particular point, or even if it is loaded at a particular point, they can mix up or solubilize with the buffer. Along with the buffer, the molecules move from one direction to the final or the other direction. In such cases, you can see this diagram. The molecule movement is not in one directional, means it is not unidirectional, it is of multi-directional. Why? Because your sample is not static, it is mostly as mobile. Means along with the buffer, they mix up with the buffer and move from one place to the other place. Now coming to moving boundary electrophoresis, here also the buffer will be loaded at preferably cathode end. Why? Because all biological macromolecules or biological samples are negatively charged in nature, hence the sample is preferred to be loaded at cathode end rather than the anode end. Why? Because they are negatively charged. Now you fill up this moving boundary apparatus first with a suitable buffer, then we load our required sample, that can be called as test sample or test solution. When loaded at one end, based on its density, they slowly move towards the other end depending on their charge. When you apply this with an electric field, the molecules move always from cathode end to anode end. Now either of this electrophoresis, whether it is micro electrophoresis or moving a boundary electrophoresis, they can be equipped with an detector and integrate the data so that you get the electrophoretic mobility of an sample from cathode end to anode end. Now this is the chart. What we see is an serum proteins. These are serum proteins where the proteins, once they are loaded at cathode end, they move towards the anode end purely based on their size. Now in this, albumins are quite greater than followed by the globulins. Next, coming to the second type of zonal electrophoresis. Here also we see three different types actually, but we'll talk about these two types because these are also outdated or which are not being widely used nowadays. One is paper electrophoresis, the other is cellulose acetate electrophoresis. Now you can see the diagram where a paper is hanged over, where one end of the paper is dipped into the buffer solution and the other will be hanging on a single string and you load your sample at one end, it moves from one end to the other end. Well, you can also see the other way of representing the paper electrophoresis. If the nature of the paper is cellulose acetate instead of a common Wattman filter paper, the type of electrophoresis under zonal electrophoresis is called as cellulose acetate electrophoresis. Now, here you can see two chambers or reservoirs where the buffer will be filled before the paper is placed. Now, once the paper is placed, you can load your samples at one end, preferably at cathode end. Now, this figure represents the movement of the molecules already. So, once you apply the electric field, molecules move from cathode end to anode end. So, this is about zonal electrophoresis. The third type, which is called as gel electrophoresis, we will discuss this in subsequent slides. So, this is about the brief of electrophoresis process. Thank you.